The DNA sequence encoding a complete V region is generated by the somatic recombination of separate gene segments. The V region, or V domain, of an immunoglobulin heavy or light chain is encoded by more than one gene segment. For the light chain, the V domain is encoded by two separate DNA segments. The first segment encodes the first 95 to 101 amino acids of the light chain and is termed a V gene segment because it encodes most of the V domain. The second segment encodes the remainder of the V domain, up to 13 amino acids, and is termed a joining or J gene segment. The joining of a V and a J gene segment creates a continuous exon that encodes the whole of the light chain V region. In the unrearranged DNA, the V gene segments are located relatively far away from the C region. The J gene segments are located close to the C region, however, and joining of a V segment to a J gene segment also brings the V gene close to a C region sequence. The J gene segment of the rearranged V region is separated from a C region sequence only by an intron. A heavy chain V region is encoded in 3 gene segments. In addition to the V and J gene segments, denoted VH and JH, to distinguish them from the light chain VL and JL, there is a third gene segment called the diversity or DH gene segment, which lies between the VH and JH gene segments. The process of recombination that generates a complete heavy chain V region occurs in 2 separate stages. In the first, a DH gene segment is joined to a JH gene segment. Then, a VH gene segment rearranges to a DJH to make a complete VH region exon. As with the light chain genes, RNA splicing joins the assembled V region sequence to the neighboring C region gene. And that's the DNA sequence encoding a complete V region is generated by the somatic recombination of separate gene segments. Have a great day.